Let's jump right into this explainer. Today we are exploring the microscopic metropolises that act as the foundational building blocks of literally all life. You know, it's so easy to look in the mirror and just see a single unified being, but the reality? It's way more complex, and frankly, so much more fascinating. You are essentially a walking, talking galaxy, composed of trillions of highly sophisticated, automated cities, all working in absolute perfect unison.
Think of our agenda as our literal blueprint for today. We're going to cover discovering life's blueprint, categorizing the cellular world, infrastructure and command, cellular logistics, and finally, core operations and scaling.
All right, part 1, discovering life's blueprint.
So, we got to start with a foundational definition. A cell isn't just some microscopic blob of jelly, right? It is the structural and functional fundamental unit of life. And the true marvel here is really that second half of the statement. A single cell is a fully self-sufficient unit. It's totally capable of executing every vital life process entirely on its own. It's an independent entity that, when scaled up, forms our tissues, organs, and eventually entire organisms.
Now, if we trace the pioneers of this microscopic world, it's really a centuries-long quest. The whole thing kicks off back in 1665 with Robert Hooke discovering the cell itself. But honestly, the picture was totally incomplete for decades. It actually took until 1831 for Robert Brown to find the command center, the nucleus. From coining protoplasm to discovering the mighty mitochondria in 1857, this mapping has been a massive human endeavor, stretching right into the modern era with the 2009 mapping of ribosomes.
And out of all those centuries of discovery came a master framework, cell theory. Pillar 1 confirms that all living organisms are built of these fundamental units, and all metabolic activities happen right inside them. But the real game changer is Pillar 2. In 1855, Rudolf Virchow dropped this groundbreaking statement, omnis cellula e cellula, the law of continuity. Basically, cells don't just magically appear out of thin air. They are an unbroken chain of life, always born from pre-existing cells.
Moving right along to part 2, categorizing the cellular world.
The crucial thing to grasp here is scale and complexity. Unicellular organisms, they're essentially one-building towns. They are primitive, sure, but they're incredibly efficient, doing all the work of life inside one single unit. They reproduce super fast, asexually, and their offspring are just identical clones. On the flip side, multicellular organisms are highly specialized, complex civilizations. Because the work is distributed across multiple cells, reproduction is slower, sexual, and results in a wildly varied progeny. Zooming in a bit more, we see a really stark contrast in how these cells actually govern themselves. Prokaryotes, like yeast, are kind of the rugged minimalists. They operate without a central membrane to protect their genetic code, relying on just a single chromosome in simple binary fission. Eukaryotic cells, like ours, well, they boast some seriously advanced membrane-bound infrastructure. They lock their master blueprint safely inside a distinct nucleus, which allows for complex division methods like mitosis and meiosis.
Taking a look at the eukaryotic blueprint specifically, there are two distinct architectural styles, plant and animal. Since plants obviously can't just get up and walk to a river when they're thirsty, they have rigid external cell walls and these massive permanent storage vacuoles that literally push their nucleus out to the periphery. Animal cells don't have that rigid wall at all. They operate with much smaller, temporary storage and keep a proudly centralized command center.
Which brings us perfectly to part 3, infrastructure and command center.
Look, every city needs borders. For a plant cell, the outermost defense is the cell wall. It's built of complex carbohydrates and acts like heavy armor, giving it mechanical strength, rigidity, and keeping the cell from bursting. Just inside that, and acting as the primary outermost layer for animal cells, is the plasma membrane. It's this thin, delicate, highly elastic layer. Its main job? Acting as border control. It's selectively semi-permeable, strictly deciding what vital goods get to enter the city and what waste gets kicked out. Moving straight into the heart of the cell, we find the ultimate command center, the nucleus. Inside, everything is suspended in nucleoplasm, which is a jelly-like transfer medium. But the real magic, that happens in the nucleoid, where RNA is actively assembled, and in the chromatin threads. These thin, tightly coiled structures are made up of DNA and protein. They are the vital messengers of heredity, and they eventually condense into chromosomes during cell division to pass on the entire master blueprint. If there is just one takeaway you remember about cellular infrastructure today, it's got to be this. The nucleus is the absolute brain of the cell. It doesn't just passively store DNA, it actively coordinates every single vital life process within the system, from transmitting those hereditary traits we just talked about to micromanaging the entire cell cycle.
But outside that command center, the efficiency of the specialized departments is just astounding. You've got the mitochondria acting as a biochemical power plant, breaking down food to release energy. The endoplasmic reticulum is basically the highway system, while the Golgi apparatus functions like a post office, packaging and shipping vital proteins all over the place. Toss in lysosomes for waste management, and in plants, those enormous vacuoles acting as water reservoirs, and you've got a perfectly self-sustaining metropolis.
Okay, let's look at how things move in Part 4, Cellular Logistics and Transport.
Transporting goods across that plasma membrane really boils down to two main systems. First, you have passive transportation, which lets molecules naturally coast downhill, moving from an area of high concentration to low concentration. It's a slow, steady process and requires absolutely zero external energy. Active transportation, though, is the exact opposite. It uses energy to forcefully push nutrients uphill against that natural gradient, from low to high concentration. It's fast, it's efficient, and it usually handles the heavy lifting for large cellular nutrients. When we look at those passive dynamics, there are two key players. Diffusion is just the random dispersal of molecules. Think of oxygen gases moving freely without hitting a barrier. Osmosis, on the other hand, is highly specialized. It strictly manages water molecules in a one-way flow, and for it to work, it absolutely requires a semi-permeable membrane at the border.
Depending on its environment, a cell's going to face three distinct osmotic gradients. Drop it in a hypotonic solution, and water rushes in, swelling the cell up. An isotonic environment maintains perfect, happy equilibrium. But in a hypertonic solution, water is aggressively drained out, causing the cell to violently shrink, a process called plasmolysis. This exact type of cellular dehydration is exactly why drinking ocean water is so incredibly dangerous for us.
Which leads us to our final section, Part 5, Core Operations and Scaling.
You know, every living entity relies on a very specific sequence of 5 operations. It starts with ingestion to get nutrients, immediately followed by respiration to burn that fuel for usable energy. That energy then drives growth and regeneration, makes reproduction possible to ensure the continuity of life, and finally powers excretion, the critical flushing out of toxic metabolic waste.
Think about how the magic of respiration actually happens deep inside the mitochondria. It takes the physical intake of oxygen and uses it to break down complex glucose. This irreversible chemical reaction releases ATP, adenosine triphosphate, which is literally the universal energy currency of the cell. This exact biochemical engine churning away right now is the whole reason you are able to take a breath at this very moment.
Eventually, of course, the blueprint has to replicate. When it's time, the cell uses one of two incredible methods. Mitosis is an equitational division. It creates two perfect daughter cells that have the exact same number of chromosomes as the parent. That's the engine behind your general growth and repairing injured tissues. Meiosis, however, is a reductional division specifically used for reproduction. It creates 4 distinct daughter cells, each holding exactly half the number of chromosomes as the parent cell, perfectly prepped to combine and create varied new life.
When you actually map this out, the stark difference between the two is undeniable. With mitosis, you have a linear, 4-stage clone assembly that culminates in 2 identical genetic copies. But with meiosis, you see a complex branching sequence that requires 2 full stages of division, splitting the genetic material down again and again to produce totally unique reproductive cells.
So, I want to leave you with a thought today. You are essentially a macroorganism piloted by an unfathomable number of self-sufficient microscopic entities that are constantly communicating, dividing, and metabolizing energy on your behalf. If your entire existence is governed by trillions of these autonomous cities constantly working in the background, how does that shift your perspective on what it means to be alive? I really hope unpacking this blueprint gives you a newfound appreciation for the incredible, unseen logistics happening inside you every single second. Keep learning, keep exploring, and we'll catch you in the next explainer.